Good morning. Happy New Year to everybody. My name is Red Schiller. I'm interim chairman of Department of Family Medicine and Community Health at Mount Sinai, and also president for academic affairs at the Institute for Family Health. And we are delighted to have Dr. Krammer this morning. Just to remind people, if it's flu season, it's time for a shot of Dr. Krammer. So we're delighted to have him update us on what's going on with flu, COVID, and other viral infections. Dr. Krammer is professor of vaccinology at the Department of Microbiology at the Icahn School of Medicine. He received his advanced training in biotechnology and applied virology at the University of Natural Resources and Life Sciences in Vienna, Austria, and completed his postdoctoral work in the Department of Microbiology at the Icahn School of Medicine at Mount Sinai. Since 2019, Dr. Krammer has been the principal investigator for the Sinai Emory Multi-Institutional Collaborative Influenza Vaccine Innovation Center, which aims to develop improved seasonal and universal flu vaccines that induce long-lasting protection against drifted seasonal zoonotic and future pandemic influenza viruses. This laboratory, which is also part of the NIH-funded Centers for Excellence in Influenza Research and Surveillance, focuses on understanding broadly reactive immune responses against surface glycoproteins of RNA viruses, such as influenza, with a goal to develop better vaccines and novel therapeutics. We are so grateful to have Dr. Krammer come back for his sixth record-breaking time to share his expertise in the constantly evolving area of medicine, and obviously the emerging public health crisis due to the resistance to vaccine acceptance. Thank you, Dr. Krammer, for joining us. And just a note, please try to complete the evaluations at the end of this rounds. Dr. Krammer?
Thank you very much, and thanks for the invitation to have me back here. I'm not sure how I feel about being associated with the flu season, but I'm happy to give an update on the situation right now with respiratory viruses, specifically with the influenza situation. But there's also some COVID-19 updates, and I'll also say a few words about H5N1 or avian influenza. Okay, so I'll just start with my disclosures. Mount Sinai is developing a number of vaccines against SARS-CoV-2 and also influenza, and I'm partially inventor on some of the IP that was filed for this, but I'm not going to talk about any of our vaccine candidates today. The objectives for today are, and this is more focused here on influenza, is to basically understand the situation with seasonal influenza and with avian influenza, specifically H5N1, right now in the US, but also globally. And so I'm going to start initially with COVID before I go into influenza. And the COVID part is going to be short, but there's some interesting developments that I think are worth knowing. So as we all know, COVID-19 had a pretty big impact on the global population. When we look back to the COVID-19 pandemic, officially we have a count of about 7 million COVID-19 deaths. Until 2023, there are pretty good also estimates that are based on excess deaths. So that's not official numbers, but estimates. And that put the number of COVID-19 deaths at around 27, 28 million, which is a significant number. But we also see that over time, the virus has made less and less impact on the general population. What you can see here is a slide that I think I might have shown last year as well. This is from the CDC. What you see in orange is basically weekly test positivity in their kind of cohorts, in their surveillance cohorts, and you see that there's an up and down of virus circulation. The virus didn't go away. But what you see in blue is basically weekly deaths. And we see a huge difference there starting basically in 2022, where the case numbers decouple from the weekly deaths. And that basically just means a lot of people have no immunity, and the disease has become less severe for the general population. That doesn't mean that it's not still a problem for people who have weak immune system, for relatively old people, for people with other underlying conditions. COVID-19 can still be a big issue, but it's not as big of a problem anymore as it was during the pandemic. So that's, I think, good news. Um, as you all know, the virus has changed tremendously over the course of the pandemic and then even afterwards. What you can see here is data from Mount Sinai's pathogen surveillance program. This is run by Harm van Bakel, and he provided that data to me. You can see here in the beginning, in 2020, we had the original or ancestral SARS-CoV-2 strain, and this is really New York City and Mount Sinai specific. We had a number of variants that showed up early. And then here, BA1, this was Omicron. Omicron, of course, made a big impact. And since then, everything that's circulating is Omicron derived, right? And right now, what most infections that are occurring with SARS-CoV-2 in the United States are based on this new variant that's called XFG. Just to let you know, whenever you see a variant that has an X, that means that's a recombinant of two different variants. So these SARS-CoV-2 viruses can recombine. And this is the current situation in terms of variants in the United States. As I said, XFG is really the dominant variant right now, this LB.8.1, and we'll get to that. That's the current vaccine strain, but they're pretty closely related. So basically, there's nothing too exciting about the current situation in the US. Globally, it's a little bit different. And what I'm showing you here, and this is also coming from the from Mount Sinai's pathogen surveillance program. This is really brand new data from right now. Is COVID-19 test positivity, right? So that's a measure of how much virus is circulating, and you have here this yellow line that's SARS-CoV-2, and right now we are really, really low. So there's barely any cases. There are some, but compared to other pathogens and compared to you know. The course of the last year, there's very little SARS-CoV-2 in New York City right now, which is good. There's also vaccine updates. Just to remind you, starting in 2022, we had updated COVID-19 vaccines. In 2022, we had these bivalent vaccines. And then 2023, we had a monovalent XBB.1.5 vaccine. In 24, we had JN1 or KP2 vaccines. And in 2025, the mRNA vaccines changed to LB.8.1, and Novavax stayed with the JN1 vaccine. And that is actually, both are pretty good choices. What we do in research now is we construct these antigenic maps, which basically tell you how closely related viruses are in terms of our serum neutralizing them. And what you can see here in this cluster up there, this is this LB.8.1 strain that's in the current vaccine, and this is XFG. This is what's currently circulating, and they're very close together, which means that this is a very good match. The vaccine matches what's circulating. Also pay attention to this gray dot over here. This is BA3.2, and we'll talk a little bit about that one. That's the new kid on the block. Also, there's already data, and this is from Columbia, from David Ho's lab, about what this year's vaccine is doing. And I'm happy to report that this year's vaccine is actually inducing very strong and neutralizing antibody responses against the circulating variants. So you can see that here. This is the vaccine strain LB8.1. In dark blue, we have neutralization titers before vaccination. In light blue, after vaccination. So they go up substantially tenfold, but they also go up against XFG tenfold. And so that's the current variant that's dominating. So this is also good news. This year's vaccine is working. Maybe also as an update, there's a new vaccine that has been licensed by the FDA recently, a new Moderna mRNA COVID-19 vaccine. The old vaccine is based on the full spike protein of SARS-CoV-2. And this new vaccine is really focusing on the N-terminal domain. So this is this green part here. and the receptor binding domain. And so they have been stitched together and are delivered as mRNA vaccine. And that was done because most of the neutralizing antibodies actually bind to these two parts and not to the gray or the yellow part of the spike. And so this vaccine has been shown to be about 10% better compared to the regular Moderna vaccine. It's not widely available yet, but I think that's something that will be available in the next season. The last COVID-19 slide is about this BA3.2, right? This is this new kid on the block. If you remember when Omicron came, right, in end of 2021, there were initially three subvariants of Omicron. One was called BA1, that was the main one. Then we had BA2, and then we had one that was called BA3, and there were very few BA3 cases. But recently, in the end of 2024, a mutated version of this BA3.2 resurfaced in South Africa. First, there were just a few cases, and actually there was not a big increase in cases, but during 2025, this virus was able to spread globally. And now this is taking over in Europe and in Australia. We can detect it in wastewater in the US, but there was not a single clinical isolate yet. But in places like Scotland, for example, it's already 10% of the circulating viruses. And the issue here is that we again have a lot of mutations in the spike protein. Again, this is increasing slowly, so that's not a big deal right now, but it could be the next variant that circulates in the ongoing year, and if we go into the next summer and winter season. So this is something to keep an eye on. Other than that, the COVID-19 situation is pretty calm. Now I want to go into seasonal influenza, and I think that is an interesting season this year, and I'll give you more specific idea of what is going on there and what could have caused this severe season. But I'll just start out with some basics. As you all know, influenza viruses are part of the Orthomyxoviridae. We actually have four types of influenza viruses, and from a clinical perspective, only influenza A and B are relevant. Influenza C can infect humans, but usually causes common colds. And influenza D only infects cows and sheep and goats. There's no human cases known. These viruses are enveloped. They have a lipid envelope. You can see a model here on the left side. Basically, this yellow layer here is the lipid envelope. Inside, we have an RNA genome, and that's made of 8 genomic segments. So you can imagine that like human chromosomes. And that's actually a problem because these viruses can exchange these genomic segments and then new viruses can emerge. One of the segments codes for the hemagglutinin, which is the major spike protein of the virus that it uses to attach to our cells. And the second one of these genomic segments codes for the neuraminidase, which is a second spike protein that actually is the receptor destroying enzyme. And so those two proteins are called HA, hemagglutinin, and NA, neuraminidase. And when you think about subtypes, they're called after that, right? H1N1 means that this virus has a type 1 hemagglutinin and a type 1 neuraminidase, and H3 and 2 has a type 3 hemagglutinin and a type 2 neuraminidase. So that's how they are labeled, right? H5 would be an type 5 hemagglutinin with an N1 neuraminidase. And so here on the right side, you can see a really nice electron microscopy picture where you can actually see the virus. And if you count the dots on the inside of the virus, you see these 8 dots, and these are the 8 genomic segments. And the fuzzy stuff on the surface here, that's the hemagglutinin and the neuraminidase. So influenza viruses are interesting because, of course, for you, this is mostly relevant because it's a human disease, but they actually have a very wide host range. They can even infect alligators. What we see in humans is a spectrum of mild to severe respiratory disease. Sometimes it's even asymptomatic. And the viruses comes in two forms. It causes epidemics. Those are caused by influenza A and B viruses, and pandemics. And the pandemics are only caused by influenza A viruses. In terms of the epidemics or seasonal influenza, we have about 3 to 5 million severe cases every year globally. and between 300,000 and 650,000 deaths globally. In the US, this can go up to 50,000 per season. So the seasons can be pretty severe. And then we have this second form, which is pandemic influenza, and I'll talk a little bit more about that later. But the strongest example for pandemic influenza is, of course, the H1N1 pandemic of 1918, which claimed approximately 40 million lives. But as I said, the virus infects a lot of different animals. It can cause infections in other mammals, and there the disease is very similar to what we see in humans, basically mild to severe respiratory disease. And the virus can really infect a lot of different a lot of different mammals. Now we also know that it jumps into cows sometimes. And we have avian influenza, which infects birds, and there it's mostly causing infections in the GI tract. Often they are asymptomatic. In some cases, and we'll talk about that when we talk about H5N1, it can also cause very severe symptoms, but often it's asymptomatic. A little bit about influenza pandemics and seasonal influenza in humans. This is a graph that I like to show. That's the history of influenza circulation in humans in the last 100 years. So in 1918, we had this big pandemic with H1N1, and then the virus started to adapt to humans better, and humans developed immunity. The virus had to escape from that immunity, and it started to change, similar to SARS-CoV-2. Became a seasonal virus, and it started to change its surface spike proteins. That's what we call antigenic drift, right? So that's why this arrow here changes color over time. In 1957, we had another pandemic. This claimed fewer lives, about 2 to 3 million, with another subtype, with H2N2. And interestingly, that new pandemic virus kicked the old virus out of the population. H2N2 then also became a seasonal virus, started to change. And then in 1968, we had another pandemic with H3N2, so yet a different subtype. And again, we had 2 to 3 million deaths globally. And again, this H3N2 subtype, this new pandemic virus, kicked out the old seasonal virus. And so H3N2 is still circulating, and that's actually the troublemaker in this season. And it's changing quite a lot over time. And that's a problem for vaccines and updating the vaccines. In 1977, H1N1 came back, was circulating till 2009, and then was replaced by another H1N1 variant, which was genetically completely different. It came from pigs. So that's the swine influenza that emerged in Mexico, and that kicked out this old H1N1. And so basically, we have H1N1 circulating in humans, H3N2, and influenza B, and they change all the time, and that makes it hard to keep up with the vaccines. And then there's this question mark here because we don't know what virus and what subtype could cause the next pandemic. And again, I'll get into that a little bit more in the end. So we have these influenza seasons, right? In a relatively regular basis. Some are more severe, some are less severe. What you see here is basically the influenza seasons in the Northern Hemisphere between 2015 and 2020. And basically, it's really every winter the viruses are coming back, case numbers go up, and during summer, case numbers go down, right? And so the question often is, where do these viruses come from? And it's basically a global circulation, right? So when our winter ends, basically the fall and winter on the southern hemisphere starts, and you start to see a lot of flu circulation there. And it's basically just a ping pong effect between the two hemispheres. It seems the virus is doing better when the weather is colder. And so that's why we have these seasons, and it's just going back and forth between the southern and the northern hemisphere. Interestingly, in the tropics and subtropics, we often have year-round circulation of influenza. So there, the weather doesn't impact that much on circulation. But it's basically just going back and forth between the hemispheres. There was an exception in terms of influenza seasons, and that was the 2020-2021 season. So the blue line here is influenza cases. This is global. And there was no season. And the reason for that was because we had the COVID-19 pandemic. There was very little air traffic. There was little travel. There were social distancing measures. People were wearing masks, and influenza is pretty sensitive to that. And and for that reason, basically that season was blunted, and there was not much circulation. But the virus did come back in the 2022-2023 season. So what you can see here, this is now Northern American US data. This blue line here is the 2020-2021 season where we basically had no influenza. And then you can see the 2021-2022 season where the virus came back, not very strongly, but it did come back. And then in the next season, we actually had a pretty early start of the season, and influenza was basically back doing its thing in the winter season. Now, this year is special for a number of reasons. The first thing was that Japan, which is basically in the northern hemisphere, reported a very strong and very early influenza season. Their influenza season started already in September, which is highly unusual. They had a lot of cases. They had to close schools. It wasn't that the cases were more severe than usual, but they had a lot of cases. And that basically led them to declare an epidemic and put in measures to kind of slow down that epidemic. What coincided with that was a antigenic drift. So the virus had mutated. And so the majority of these infections were caused by H3N2. And a clade that was called J.2 was circulating, and that had changed. There were a number of mutations that had occurred. And then this new virus, this drifted virus, was called subclade K. And you see this on the right side. This is a structure of a hemagglutinin protein. And then you see major changes. This is this original virus, hemagglutinin. Here on the right side, you see two key changes. And what these changes do is they allow the virus to circumvent our neutralizing antibody responses to some degree. And that made the virus more fit. And that was the reason why there was all of a sudden more circulation. So this started in Japan already in September. Then we heard from the UK that they had an early start into the season. Their season started already in October, which is also very unusual to have such an early season in the UK. Basically, it went slowly, but then it hit the rest of Europe. So Europe had a little bit of an earlier season, and now it's hitting the US. And the interesting thing in the US, it's not necessarily an early season, but it's a relatively severe season. On the left side, you can see basically the deaths that CDC is evaluating and looking for the type of influenza circulating. So it's almost exclusively H3N2. That's the red bar here. There are some other subtypes circulating too, but it's mostly H3N2. And then here on the right side, you see the this year's season, basically the red line, and you can see that overall it's not starting earlier for the US, but it's really a lot of cases, right? We're already exceeding the numbers of cases for the last few seasons. So it's a very strong season. And when you look at across the US, so that's the latest CDC data, you see that in most states, the activity is either very high or high. So again, very strong influenza season. I think the positive part here is that New York City is already on the decline. So it seems that the virus infections peaked here. This is the graph that I showed earlier. You can see here, this is the peak from last year's season, right? Again, this is test positivity, which gives you an idea of how much virus is circulating. And then this year's season in New York started a little bit earlier, but not drastically earlier than usual. And we had this really high peak here in the end of December. You probably have seen many influenza cases during that time, but the good thing is we're going down. And if that trend holds and it goes down that steeply, that wave should be over relatively soon. How well do the vaccines work this year? The bad news is the vaccines don't match. The H3 strain in the vaccines does not match the the H3 strain that is circulating. That doesn't mean that the vaccine doesn't work, but it might not work as good as it would work if there is a match. But because the season started so early in the UK, they were able to measure vaccine effectiveness pretty early on. So we have some data on how well the vaccine works. You have to take this with a grain of salt because, of course, they started to vaccinate in September, and then in October their case numbers went up. So they evaluated their vaccine effectiveness relatively soon after vaccination, and of course, then the vaccine works better. The other important thing to keep in mind here is when we look here at the age group 2 to 17 years, we're talking about the intranasal flu vaccine, which the UK uses for kids. When we are talking here about older age groups, then we're talking about the injected vaccine. So it's two different vaccines. And here, basically, you have emergency department attendance and hospital admissions, so two different measures. And what they could measure in the UK was that the intranasal vaccine for kids works really, really well. So the vaccine effectiveness is around 70% against both emergency department attendance and hospital admission. And the vaccine also works, even though not as well, in the older populations. So here we have vaccine effectiveness somewhere between 30 and 60%, which is actually not so bad for influenza, specifically when it's mismatched. And I would like to take that opportunity to kind of point out the difference between vaccine efficacy and vaccine effectiveness, and maybe also talk a little bit about why we often see these lower numbers for influenza. So if you remember when the SARS-CoV-2 vaccines came, the numbers were super high, right? It was vaccine efficacy of about 95%. That was exciting. And that was measured in a phase 3 study or in phase 3 studies where we compared the vaccine with a placebo control in a population that is completely naive, that has no immunity to the new virus that's coming in. And this is done under ideal conditions because in a phase 3 study, everything's controlled, right? So you get the vaccine efficacy, which is a relatively high number. Now, for influenza, we usually get the vaccine effectiveness, and that's different. That looks at how well does the vaccine work after rollout in the population. And you cannot measure that with a placebo control group because you don't have that, right? So this is done with a test negative design. And the problem here is that the control group already has partial immunity because a lot of people have exposure to influenza from, past vaccinations, past infections. And so that's why the number for influenza is often only 50%. And that's not that low. If you would compare it to a naive control group, the number would be much higher. And I think that's also important to tell patients when they ask about the influenza vaccine. And if you hear about numbers of 30%, that sounds low, but that also has to do with the way it's assessed. And the other point that I wanted to make here, and I'm using an old advertisement from CDC that they're not using anymore. One important point with the flu vaccine is not necessarily to stop infections, but to basically attenuate the infections, right? To make sure that people don't progress to severe disease. And I really like that advertisement. Again, they don't use it anymore, but I think it's good for messaging. And so I'm going to switch now to the last part of the presentation, and that's avian influenza. And so I showed you this graph earlier, and I pointed out pandemics, right? H1N1 in 1918, H2N2 in 1957, H3N2 in 68, and then again H1N1 in 2009. And so the main question is, where do these viruses come from? For influenza viruses, we have a vast diversity of subtypes. So for influenza A hemagglutinins, right, we by now have, this is actually, I think this is correct. There are 19 different subtypes, right, that circulate in animals. We know, and this is indicated by the red stars here, that H1 and H2 and H3 can cause pandemics, and some of them are circulating right now in humans. The blue stars indicate subtypes that have caused zoonotic infections, so where people got infected by interacting with animals that had these influenza virus strains. And then we have a bunch of them that have never never infected humans, but potentially could, right? And the same is true for the NA, for the neuraminidase component. There's many different neuraminidases. And all of these viruses circulate in animals. The main reservoir is aquatic birds, but the viruses also spill over into marine mammals, into bats, into other birds, into domesticated animals, and of course into humans, right? So there's this vast reservoir. And that is the problem because any of these viruses in this vast reservoir could jump into humans. And this happens every now and then. I like to show this slide. Um, Basically, what you have here is a timeline of recorded infections, zoonotic infections, where people got infected with avian influenza viruses or other mammalian influenza viruses. So this happens all the time, and often it's reported. Many cases, probably it isn't reported. This can have drastic consequences for the infected individual. These viruses can cause very severe infections. In other cases, the infections might be relatively mild. And I'm showing this picture here because what we have seen repeatedly is actually infections where swine influenza viruses jump into humans at country fairs in the Midwest. This is well documented, and this is also a potential source for a new pandemic. And I wanted to talk about a specific virus today, and that's H5N1. I mentioned this, and I think I talked about it last year as well. H5N1 is one of these avian influenza virus subtypes that for a long time wasn't seen as a threat for humans. It was something that occurred in poultry operations. It was special because it actually does make birds sick and it kills chickens within a very short period of time. But it was seen that this is not a problem for humans. This all changed in 1997 when there were cases in Hong Kong. Actually, they detected 18 cases. These people infected themselves when they went to a market to buy live chickens, and 6 of them died. And so since then, people are afraid of H5N1, and it is seen as a pandemic threat. And since, 1997, the virus has started to circulate mostly in Asia, in Europe, and in Africa. It also has started to diversify quite a bit. These viruses mutate too. They go through antigenic drift. And right now we have two major clades that we are worried about. One is actually what we see now in North America and in the US, and that's called clade 2.3.4.4b. And then if you hear about cases in Cambodia or Vietnam, those are different clade. They are clade 2321E. You don't have to remember that one, but the clade 2344B is important because that's now circulating globally. So this virus showed up in 2020 in Europe. Probably initially came from Egypt. It was a very fit virus in wildlife, in birds. And so it started to circulate all over Asia, all over Europe. And then in 2021, also it made it to North America via migratory birds, right? There's birds that fly from Europe, often via Iceland, to North America, and they can carry the virus. Some species of wild birds, they get infected, but they don't get really sick. They can still fly and they transport the virus. And so, as you can see here, through 2022, this virus spread throughout the Americas, and it now also reached Antarctica. And it also started to reassort. So it started to mix with avian influenza viruses that are present in North America that are less problematic. And so now we have H5N1, but we also have viruses like H5N5. And the recent human case, the human mortality or fatality in Washington State, that was an H5N5 virus. And so along the way, it hit a lot of different bird populations, and it's actually a problem for conservation because it hit bird populations that are critically endangered. And so it, for example, hit the Californian condor population. And so researchers started to vaccinate these birds because there's very few of them left. And if they die of avian influenza, they're going extinct, right? We also had a lot of die-offs in the Antarctica, really mass die-offs where millions of birds died. So it's a huge problem for conservation. The problem is also that when these birds die or when they are weak, they get eaten by foxes, for example, by raccoons. basically mammals that feed on these carcasses can get infected too. And we had a lot of these cases in the US. And the problem is when that happens, of course, the virus starts to replicate in these mammalian species, and it's an avian virus, but it starts to adapt to the mammalian cells, and then that can become problematic for humans too. And so this is just a map across the US where mammals have been found that were infected with avian influenza. And typically, these are dead end infections. These animals die and they don't spread the virus to other mammals. But there's exceptions to that. We have seen spread of this avian influenza in fur farms in Europe and also in marine mammals in the Americas. And this is just one example. This is from Argentina, where elephant seals got infected, and the virus started to jump from elephant seal to elephant seal. That's a problem for these populations because these are also critically endangered animals. And their mass die-offs where, thousands of them die. But of course, when it jumps from mammal to mammal, the virus changes and it adapts to mammals, and then it becomes dangerous for humans too. And so we had these outbreaks all along the coast in South America, but we also have them in New England, for example. So there was an outbreak that has been reported here locally, where we also had H5N1 in local seal populations. So that's really problematic. And what we see right now is that the case numbers are going up again in wildlife. This seems to happen every winter now, and it doesn't seem like the virus is disappearing from North America. One important thing that should also be mentioned is that the virus made it into dairy cattle. That's interesting because influenza A viruses were not known to infect cattle. But now we have a relatively large outbreak in dairy cattle. The virus made it 3 separate times into the dairy industry. And interestingly, there it's not a virus that's transmitted via the respiratory tract, but via milk, via milking equipment. And you can actually find a lot of virus in unpasteurized milk. Um, this was, as I said, is a large-scale outbreak. A lot of dairy farms across the US were affected. Right now, there's only one active outbreak, and that's ongoing in Wisconsin. But in total, more than 1,000 farms across the US were affected. And important to know is also that this virus can go into pets. It is pretty deadly for cats. Cats that prey on birds that are infected can get infected and can die of the infection. And it can also infect dogs. And there was recently a report in Canada where a dog got infected with H5N1 and I believe died of the infection. So just to kind of wrap that up with the human situation, there have been transmission events of H5N1 to humans. In the US so far, we had about 70 cases. This is typically when humans interact with either infected birds or infected cows. And interestingly, historically, the case fatality rate for H5N1 across the globe was about 50%. But what we see now is that most of these human infections are relatively mild. Sometimes there is some respiratory symptoms involved. In many cases, when the infection is from dairy cattle to humans, it's conjunctivitis. The reason for that is that basically splashes of milk that contain virus end up in the eyes, and the virus starts to grow there. Influenza viruses can do that. But we also know of at least 8 severe cases globally. And in the US, we had one death in Louisiana. We also had one death in Mexico and one in China. And we also had a human death in Washington State very recently that wasn't with H5N1, but it was with a virus that is a resortant of H5N1 and the local avian influenza strain. And that was an H5N5 strain. There's also some cases where we can't prove that there was contact with infected animals. Those occurred in Missouri, in British Columbia, and in California. And that's, of course, interesting to follow because there is suspicion there is that maybe there was already transmission from one human to another. And that would be problematic because if that happens, this could start a new pandemic, right? So far, these avian influenza viruses are not very good at spreading from humans to humans. But if the virus acquires this ability, then this could mean that a new pandemic is starting. And the biggest problem there is not that the avian influenza virus per se mutates and gets so much better in terms of replicating in humans, but typically what happens before a pandemic is that one of these avian influenza viruses mixes with a human influenza virus. And I told you earlier that these viruses have these genomic segments, these chromosomes. And if a person, for example, is infected with an avian H5N1 virus and a seasonal, let's say H3N2 virus, then these viruses can exchange genomic segments and a new virus can emerge that maybe now has the H5 spike protein, to which humans have little immunity, but it might have other genomic segments that allow the virus to grow very well in the human respiratory tract. And that's kind of a recipe for disaster, and that could actually lead to a pandemic. So these co-infections are really what we need to keep an eye on. And I just wanted to stress that in 1957, in 1968, and in 2009, these pandemics were caused by viruses that had mixed. So these were these reassortments between a human virus or a swine virus and then an avian influenza virus. We suspect that the same happened in 1918. We can't just prove it because there's not enough data. But that seems to be the mechanism how these pandemics start. Um, just two slides about countermeasures before I wrap up. First of all, there's a number of antivirals that have been developed for influenza that are in clinical use. They do work against this H5N1 virus in vitro, in cell culture. We see that they are effective. They also work in animal models. Here, there is some data that the cap snatching inhibitors that would be Xofluza, so baloxavir, works better than the neuraminidase inhibitors like oseltamivir. So that's something that, there's no human data for that. But in animals, at least, there seems to be an advantage of baloxavir over oseltamivir. Vaccine strains have been made for these viruses, and I'll show in the next slide a little bit more about these vaccines. And seasonal influenza vaccine production could be changed very quickly to basically make these H5 vaccines. That would just be a strain change, so that wouldn't take a lot of time. But we also know that these avian influenza vaccines are usually not very immunogenic in humans, and so you would need to give at least 2 shots, not one, as we do it for seasonal influenza. And it would probably also require an adjuvant, like the adjuvanted influenza vaccines that we already have, like Fluad. I also wanted to stress that masks work pretty well against influenza, including H5N1. So that's certainly also a countermeasure that can be used. The last slide here about H5 vaccines. Historically, a lot of H5 vaccines have been developed and licensed by different licensing agencies, including by the FDA. So there is licensed H5 vaccines in the US right now. They're not licensed for the general population, but for example, if you're a researcher working on H5N1, you can get these vaccines. The problem is that there's only 2 vaccines that match the current H5N1 strain. One of them is made by Seqirus, and that's based on the Astrakhan strain from 2020. So there the H5 is very similar to what is circulating right now, even though the NA is mismatched. And this vaccine is used in Europe in several countries. They vaccinate veterinarians, they vaccinate farmers, and so on and so forth as precaution so that they don't get infected with H5N1. And then Canada also has access to a vaccine that is made with an H5N1 strain that was isolated in South Carolina. The US currently doesn't have a vaccine stockpiled that could be used or that is licensed. But as I said, this could be acquired quickly, or these vaccines could be made available very quickly through the regular vaccine production process. And so this brings me to my conclusions. With H5N1, it's becoming more and more active again. We already had 2 recent detections again in New York City. So we have a surveillance program here to look for that. So we see the virus in wild birds again. A reassortment between H5N1 and the seasonal influenza virus strains may cause a pandemic. You never know. This is hard to predict. Vaccines are available in Canada and Europe, but could also be produced quickly for the US. We know that currently used antivirals likely would work for H5. It's not very likely that a pandemic is imminent. I don't want to cause any fears here, but this is certainly we need to keep something we need to keep an eye on. And the truth is that this virus so far caused huge ecologic and also economic damage in the last 5 years. And the problem is it's not going to go away. So with that, I'll stop here and I'm happy to answer any questions.
Thank you, Dr. Krammer. What an exhaustive summary of the challenges that we face with influenza. So let's see, you can open this up to questions. I guess if I can start, what sort of, given the fact that nature seems to be so creative in just being able to challenge our efforts at, managing these illnesses, I guess are there any thoughts about different strategies, of sort of, because it seems like, we base our vaccines on what happens in the Southern Hemisphere a few months earlier. And, were there any thoughts about, different interventions that, either with new technologies that might help us get ahead of dealing with, the reassortment issues? You raised so many issues that, are challenging our ability to have the proper match between the strain and the vaccine.
There's actually a lot going on. I could have continued for another hour with new technologies and exciting developments. There's actually a number of them right now. So on the one side, one issue for the vaccines that we use right now for seasonal influenza is that we have to pick the strains in February for the next season, right? And that's before the Southern Hemisphere season even happens. So we don't know if something changes there, we can't even incorporate that, right? That's a big issue. And so this the reason for that is that with the technology that we have right now, which is really old-fashioned, it takes a long time to make the vaccines. Now, mRNA vaccines have been developed for seasonal influenza. They have been shown that they work in phase 3 trials and they can be made much faster. So the strain selection for those vaccines would have to be made in June, which is much closer to the season. So an mRNA vaccine would probably this year contain the subclade K because that was already known at that point to circulate, right? The issue right now is that, of course, there is not a lot of appetite for getting an mRNA vaccine. There's questions about would the FDA even license one of them right now, and so on and so forth. So, but that could be a potential help with matching these strains better. The other super exciting development right now, and this is more for people who have underlying conditions and cannot develop immunity by themselves that well. There's a new drug that has shown really good promise in preventing influenza, and that's called CD388. And that's basically a combination of an antibody with a neuraminidase inhibitor. And they did a phase 2b trial with that last year and showed 75% vaccine effectiveness or effectiveness. It's not a vaccine, right? So this is something that is given before the season. It's has a very long half-life and protects throughout the season. And that is a drug that was developed by a small pharma startup. The company now got acquired by Merck. And it stands to reason that this is something that's on the market in a few years. And that is not strain specific. That would even work against H5. So that's a super exciting development. But then again, there's a lot of things that can be done with doing better surveillance, improving surveillance systems, getting to know earlier what's happening. In terms of, keeping H5 or avian influenza at bay, the problem is always the interaction between humans and infected animals. So the wild animals, wild birds are not so much of an issue because typically you don't go to
Mhm.
Central Park and pet the goose, right?
Yep.
So you stay away from these animals anyways. The problem is domesticated or farm animals.
Mhm.
And there, we could introduce vaccines to protect them, and then we don't get exposed. And that's what some countries like France or China are doing, right? The US doesn't do that right now. But the challenge that I see is that a lot of surveillance efforts, a lot of coordination with the WHO, all these things that are needed to do this well are actually, reduced. And there is not a lot of interest from the government right now to kind of, keep these activities up. And I think that's a major challenge that we are
Yeah.
facing.
There are actually a couple of questions about the effects of disempowering ACIP and policies about flu and, obviously all vaccines. I get, you've expressed your concerns. I don't know if you have any advice or suggestions about what um, that is going on that try to challenge or provide recommendations that are more local. And I know professional organizations are making recommendations. I don't know what your thoughts are on that and what any suggestions that you have for those of us in primary care.
I do have, so again, I'm not a physician, I'm a vaccine
Mhm.
researcher, but I know the people who were on the ACIP a year ago, they were really, really good physician scientists. They knew what they were doing. In my opinion, the ACIP recommendations as they stood a year ago should be the ones that should be followed. A lot of medical associations are also saying that they're maintaining the recommendations as they were. And I think it's better to follow those guidelines right now or the guidelines of that New York State or California, for example, are developing as compared to what comes out of ACIP. I personally, and I'm saying this bluntly, I would not trust anything that comes out of
Sure.
the current ACIP.
Yeah. No, I think that's true with a lot of recommendations from our federal government. It's unfortunate. Um, it sounds like research on universal flu vaccine is sort of stopped, and or is it continuing and sort of maybe update? I know that was an interest of yours of just having something that basically it doesn't it anticipates or responds to the shifting antigenic challenges.
No, no, that research continues. Believe it or not, we're going into a clinical study right now
Oh,
with an mRNA-based universal
well,
flu vaccine that's still supported by the US government. So I think with influenza, at least with vaccine development, there's still interest to support that. I think for COVID-19, there's not a lot of support for vaccine development anymore. A lot of efforts are stalling there. With influenza, it's getting more difficult, but our clinical development and preclinical development is still ongoing, and so is development from other players in the field that develop these vaccines. And this drug that I mentioned, this CD8 388, in a way, you could see that also as a vaccine.
mhm.
A universal vaccine. Maybe not for the whole population, but for population parts of the population that really need that kind of protection. So I don't think these things are stopping. Things might
Good.
slow down a little bit, but I don't think we're stopping because the need is there. And the truth is there's also other funding bodies. It's not just NIH that supports these things.
Yeah, no, I see that. That's good news, and we'll hope that that funding continues. There's another question about a potential second wave of influenza. Um, Given our history, is that the peak that we saw in late December and with the drop? Is it possible there could be a second wave of influenza A?
Yes. So we had a recent season where we had a double peak. I forgot now which year
Oh,
it
yeah, yeah.
was.
That's right. Yeah.
And you have to keep in mind that this was mostly H3 and 2. There could be a second wave that is caused by H1N1. But what we often also see is a late wave with influenza B. So sometimes you see that even out in March and April. And influenza B, although it's B and not A, isn't less severe, right? Specifically in kids, you often see a lot of severe infections with influenza B. So that could still happen. And specifically, if you have an earlier season, I think we're prone to have a second peak.
Mhm.
That's often not as drastic as the first one in terms of number of cases, but it's something to keep an eye out.
There's another question about the data from Europe regarding intranasal vaccine um, effectiveness in children. Is there any data on comparing that to injected?
So there's no direct comparison from the UK just because they have pretty interesting and tight recommendations for who should get which vaccine, right? And so the majority of their children and youth are really vaccinated with the seasonal vaccine, so they don't have much to compare it to. And I would guess that the injected vaccine wouldn't work as well, even in that age group. The reason for that is these intranasal vaccines have the advantage that they do two things, right? They induce adaptive immune responses, antibodies, T cells, partially also locally in the nose, right? But because it's a replicating virus, it's an attenuated vaccine strain, you also get innate immune responses in the upper respiratory tract. So even for a few weeks, you might get relatively unspecific protection against any kind of virus infection, right? So these vaccines can have that effect. And so I think for kids where they really work well, they're better than the injected vaccines.
Mhm.
And the nice thing is, right now in the US, last year the FDA licensed these vaccines to be home administered. So actually now what parents can do in most states, not in all states, they can actually order the vaccines from a pharmacy and then vaccinate their kids themselves, which removes the barrier too. But I think for kids, these are really good vaccines.
Hmm. That's interesting. Self-administered vaccines is, I mean, as more and more of our healthcare system, shifts to certain home-based interventions, it's an intriguing um, it's an intriguing advance. There's a question about influenza B, and is that something that we need to worry about for this particular season? And
That's always something to worry about.
yeah,
Again, influenza B often causes its separate peak later
mhm,
in the season. Not every year. Sometimes it's coming also early and together with influenza A, but you see years where it comes late and on its own. And again, this can be problematic. It's from a severity of infection perspective. Over the years, the ranking is that age 3 and 2 causes the most severe infections, followed by influenza B, followed by H1N1. So influenza B is something to be really careful about, specifically in kids.
mhm. There's a couple more questions. One is somebody asked about the World Health Organization proposing only N95 masks for influenza protection. Is that something that you think that we should be using in our clinical settings? Just N95 or another controversial question that.
It's not so controversial. I think so. Some of the influenza transmission is via droplets, which are larger, and the surgical mask is going to protect you to some degree
Mhm.
from that. But a proportion of influenza infections are caused by aerosols, and a surgical mask is not going to help you against aerosols. So, just from my personal perspective, I looked at the case numbers here in New York, and in the last few weeks, whenever I had to take the subway or the bus,
Mm,
or when I walked through the hospital. I was wearing an N95 because that's something that certainly protects you from
mhm.
influenza. So
Right.
I would recommend that over a surgical mask.
Yeah. Just given the variations in immunity, I mean, it's a really good point. It's just masks do provide that kind of protection. So,
They do, and they work better against influenza than against SARS-CoV-2. So they are not, right? The number that tells you how infectious the virus is, how many people an infected person infects, it's much lower for influenza. It's around 1.5. So if you put up a little bit of a barrier that usually does the job, for SARS-CoV-2, it's much harder.
Mhm, mhm. And finally, could you repeat what you said about influenza in tropical areas? Like, just somebody wanted to hear that again.
So in the northern and southern hemisphere, we really have these winter seasons, right, where we have a lot of cases. But when you look at places like Nicaragua, for example, right, you have waves year round. Often they coincide with rainy seasons, but not always. And so it seems that in the northern and southern hemisphere, the season is really regulated by colder temperature and maybe also the fact that people spend a lot of time inside,
Mhm,
which kind of drives infections too. While in the tropics or subtropics, it seems to be a year-round phenomenon, and you have year-round waves.
mhm. Great. Well, it's 9 o'clock. Dr. Kramer, just an exceptional wealth of information, and we're always delighted to have you and look forward to next year and sort of newer challenges. Thank you, everybody. Happy New Year, and have a good weekend.
Thank you. Happy New
Thank
Year.
you. Yeah, take care. Fill out the evaluation, please.